survey of bacterial diseases, part 1. This will be 3 lectures. So we will do 1 lecture this week and 2 lectures next week. There is a mistake on the syllabus. It says that this is chapter 10 and 11. Chapter 10 is actually not being covered during this time. It'll be covered later in the class. But I have some material from chapters 11, 24, 25, and 26, as well as supplemental information, mostly from CDC. And so you'll see that as we look at different examples of bacteria from different phyla, that I will have a lot of specific information about diseases that you don't find in your book. Okay, so as I give you information, you definitely want to take down notes on everything that I say, because you won't necessarily find it in your book. All right, so we will start with a respiratory review, brief review of the respiratory system, the gastrointestinal system, the urogenital system as they pertain to infection. Then we will dive into the first part of our bacterial survey, which will be the Proteobacteria. And then in lectures 2 and 3, we will pick up with firmicutes and work our way all the way through. So the Proteobacteria are the largest group, and that's why we're going to stop there for this lecture. Okay, so phyla is the plural form of one phylum. And also want to give you a heads up that I know in the first lecture I made you memorize do keep pots clean or families get sick. But when we're looking at bacteria, we're not even going to see kingdoms. It's going to go straight from domain to phyla. Okay? All right. So here's our brief anatomy review as it pertains to infection. So structures of the upper respiratory system that you need to remember are the sinuses, the nasal cavity, and of course you know that the sinuses are both above and below the eye, and these are hollow cavities where air can flow from the nasal cavity where it gets warmth and moisture, but also very importantly from the perspective of infection. As air passes through the nasal cavity, it's you get the first filtration layer as air travels through the nose hairs and whatever mucus might be in your nose. That's going to catch some of the debris in the air, so that acts as like a filtering effect. And then of course, as it passes through the nose, it's also being warmed and moistened. As it goes through the nasal sinus cavities, it will continue to be warmed and moistened, and then it is ready to go into the lungs. Okay, so the upper respiratory system stops at the lungs, right? So the lungs are the lower respiratory. So you have to also make sure you know that, of course, the oral cavity, the tongue, those are also part of the upper respiratory system, as well as the ear. And when we talk about ear infections, we would really be talking about the middle ear or the auditory tube, which is a drainage from the ear to the throat. Okay. And then we have the epiglottis, which has a very important function of closing off the airway, which you see in pink, so that when you swallow food or water, it goes down the esophagus, which is in the back here. Okay, so if you've ever said like, oops, I swallowed the wrong way, I'm choking on my water or whatever you swallowed the wrong way, that would be a failure of the epiglottis to close off all the way. directly underneath that is the voice box called the larynx. And as air passes through the voice box, it vibrates what we refer to as vocal cords, and that's how you get different sound. So however tight the vocal cords are being pulled, okay, you have muscles that surround this whole area. So however tightly you pull those muscles will change the pitch that is produced as air vibrates those. So again, I know you learned anatomy already, but now you want to focus on anatomy as it pertains to protecting against infection, and then when you actually get an infection. So anatomical defenses of the immune system of the upper respiratory system include those nose hairs, like I mentioned, that filter the air, the mucus, which is, throughout the nasal cavity, mouth, throat, everywhere, okay? And you also have IgA antibodies, which are found in mucus, and that would specifically target specific pathogens that you've encountered before. And then you have lymph nodes that are, you have bundles, pouches of lymph nodes in your tonsils, right? And of course, that's strategically located so that as you breathe in, whether through your nose or through your mouth, it will pass by your tonsils, or as you swallow as well. Okay. We also have the ciliary escalator, which really applies to the lower respiratory system, which is lined in cilia and constantly propels mucus up and out of the lungs and drains into the throat. And then in the lungs, you also have alveolar macrophages. And then in your upper respiratory system, you have the microbiota, such as in your mouth and nose and your eyes, but your eyes are not part of the respiratory system. Okay, so I started to talk a little bit about the lower respiratory system, so we're go ahead and take a look at that. But real quick, just to point out, so the tonsils right at the back of your right, kind of the back of your mouth, the beginning of your pharynx, your throat. Okay. So structures of the lower respiratory system are going to include your windpipe, right, called the trachea, your lungs, and your lungs are made up of smaller and smaller branches, right? So from the trachea being the largest branch, then smaller and smaller until you get down to single cell level of what we call the alveoli. And the alveoli are these little balloon-like sacs, if you recall, that are a single cell thick. And that's where your gas exchange occurs, very importantly. So that's why you see all these blood vessels wrapping around the outside of the alveoli. And this is where we focus attention when it comes to if you have like pneumonia, for example. If you have bronchitis, that would be your bronchus and your bronchial tubes, so those larger branches would be affected. And if you had pneumonia, it'd be at the alveolar level. We'll go ahead and look at that in just a second. I've included a couple short these videos are long, but I've told you where to begin watching so that you don't have to watch the whole video. Just to give you a little bit more review of the structure and function of the respiratory system. The lower respiratory system should be free of microbes, but we do have, as I mentioned, the alveolar macrophages that crawl around and search from alveoli to alveoli, just in case any bacteria or viruses or fungus have made it all the way down into your alveoli, you would take care of it, but your alveoli macrophages would take care of it. Okay. The idea of the normal microbiota of your upper respiratory system is also to help prevent pathogens from getting into the lower respiratory system. But those, the mucus, the ciliary escalator, very, very important. And then, of course, the alveolar macrophages. So you can imagine then if you are a microbe trying to cause an infection, you would need to be able to evade all of those defenses. Okay. So if you have an infection, it would trigger inflammation typically. And so a lot of our terminology then ends with itis, I-T-I-S. So pharyngitis, laryngitis, tonsillitis, sinusitis, epiglitis, and otitis, right? So that all means inflammation of, and then whatever precedes it is telling you the location. So when you have inflammation of the throat, it's pharyngitis, and that would be a sore throat because of the inflammation. It may or may not include swelling of the tonsils, right? But the mucous membranes of the throat are inflamed. You can have more symptoms. You may or may not have more symptoms. Okay, laryngitis, when you have inflammation of the voice box, typically you lose your voice, or you would have a very raspy, weak voice. You typically don't have any other symptoms when you have laryngitis. It's very targeted at the voice box, but you could have some fatigue. Tonsillitis, inflamed tonsils, sometimes is accompanied with full-blown pharyngitis, fever, etc. And other times people have recurrent bouts of inflamed tonsils that aren't really associated with fever or any other cold or flu-like symptoms other than the swollen sore throat. Sometimes people have such recurrent tonsillitis that they will end up having their tonsils removed. Typically, as you become an adult, your tonsils will get smaller and smaller, and some people, they're completely undetectable. Sinusitis, a lot of people think they have sinus infections when they just have a normal cold or any ailment. They think if they have congestion that they have a sinus infection. Sinus infections are actually not too common, and furthermore, there's a debate whether or not to treat them. But anyways, so the cavities of the cranial bones that open into the nasal cavity get filled up with thick mucus, putting lots of pressure and pain on the face, on the teeth, right? But it is usually self-limiting, meaning that it will resolve on its own. If the pain is severe or continues long enough, then it might be treated with antibiotics. We really want to limit the frequency of antibiotics to kind of try to control at least the rate of antibiotic resistance. The epiglitis, on the other hand, if that were to become inflamed, could be life-threatening, cut off your airway. So we actually have a vaccine which is given to babies to prevent a particular infection called Haemophilus influenzae, which is a very confusing name because it's actually not the influenza virus. This is actually a bacterial name, and that's its unfortunate name, but sometimes it's referred to as Hib because it's a type B. And so this is a bacteria that, as I said, can infect the epiglottis, make it very swollen. It can be very, very sudden, and with just a few hours before you have any idea that anything is even wrong, it can cut off the child's airway, and of course that would lead to death. So children are the most prone to this, and like I said, we do vaccinate for this. Children are also the most prone to ear infections, otitis media, and it's usually the result. This is usually a secondary infection, complications of either pharyngitis, tonsillitis, sinusitis, right? So complication of a sinus or throat infection. You don't usually start with an ear infection with no other symptoms. And this is also the topic of a lot of debate, whether or not to give antibiotics to treat ear infections. I think it used to be given because, well, not only is it quite painful and babies and children will cry and cry, but there was, of course, always that fear of, could this lead to any type of hearing problems? But as more research more data has been collected on this matter, it doesn't seem like there's any risk of not giving these antibiotics. But there's lots of risk of giving, over-prescribing antibiotics. So another thing is that for some children, they're more prone to these ear infections because those tubes that drain from the ear to the throat, sometimes they are not as vertical as they ought to be, so they don't drain as well, and they get more infections, or they get infections more easily. But typically that resolves itself as the child grows and they become more vertical. But sometimes surgery is performed to correct that. Respiratory infections overall are the most common of all types of infections, and that's not too surprising given that you're putting upper and lower respiratory into one category, and the air, as we already learned, is full of microbes, bacteria, viruses, fungi, everything, you name it, and we are constantly breathing it. Okay, so even though we have all these mechanisms in place to prevent infection, they're still the most common of all infections, and GI tract would be the second most common, which is also not surprising given that we're always eating and drinking and ingesting microbes into the body. All right, so some good stuff starting. Okay, another thing I want to point out is that there are some really clever, interesting pathogens that start or enter into the body through the respiratory tract, but then actually travel to some other area for their actual colonization of where they want to infect. Examples of this would be mumps, measles, and rubella, right? You've seen those rashes, right? And so you know that they don't stop in the respiratory system, they continue on. All right, lower respiratory. The two main conditions that we want to talk about are bronchitis and pneumonia. They are two completely different conditions. Bronchitis and pneumonia are general terms that indicate inflammation in a particular region. Pneumonia specifically means water or fluid buildup in the alveoli. Okay, so bronchitis is an inflammation with mucus in the bronchial tubes, and that inflammation in that mucus has then made a narrower pathway, and it is harder for air to get in and out of the lungs. Any microbe, any pathogen can lead to bronchitis, right? Bacteria, viruses, fungi, and even sometimes allergies can cause this as well. Okay, pneumonia can also be caused by any of bacteria, viruses, or fungi. Usually it's either a virus or a bacteria, but fungi can cause infection as well. So with bronchitis, typically you'll have cough with a phlegm. So you cough up phlegm, you have a wet cough, and it will last for more than 5 days. You'll typically have a sore chest and be very tired. And the entire illness can last several weeks. If breathing becomes really difficult, you would be given some type of inhaler to help open up those airways. Of course, if you ever have difficulty breathing, you should go straight to urgent care or ER. And if you ever develop a cough with blood, that would also be reason to go straight in. And if your fever is too high, or you've had a prolonged fever for, more than 3 days, those are also reasons to go and see the doctor. Okay, so one last thing to talk about with bronchitis, this inflammation ends up damaging the tissue of the bronchial tube, and so that's what this image here is showing, some damaged cilia, and that leads to the buildup of the mucus. So with chronic bronchitis, the lining may stay inflamed and the cilia may not function at all, and then you get an even thicker buildup of mucus, and so it can be quite problematic. And also this is similar, not the same cause, but similar idea to what we're seeing with cystic fibrosis, but there is a difference. So pneumonia, you typically also have a cough with phlegm, and this phlegm will be green or yellow, and it can also be bloody. And you typically have quite a fever, anywhere from mild to high fever, and it may fluctuate between the two. You may have shaking chills with that fever, and as the fever goes from high to low or breaks, you could have excessive sweating and clammy skin. You can have a headache as well, and sharp stabbing pain in the chest that becomes worse when you try to breathe deeply. And if you look at this image here, you can see that there's fluid in the alveoli, and that makes it wherever the fluid is, you're not going to have gas exchange. So of course, trying to take a deep breath is just going to it's going to hurt because you can't do it, essentially. Okay, so you're going to become winded easily. It could be as simple as just trying to walk to the restroom, depending on how severe the pneumonia is. It can be just hard to do everyday things. Don't ever tell yourself, if you or someone you know is having difficulty breathing, and they say, oh, I just have pneumonia, I just need to rest, you get them to the doctor and make sure that they've been evaluated and told what to do by the doctor. Okay. You typically have a loss of appetite with pneumonia, very low energy rate. It's hard to breathe. Of course, you're going to be tired. And if that's allowed to continue long enough, or if it's severe enough, if you're not getting enough oxygen or enough food, confusion can happen. So, fever and not enough oxygen is a bad combination. And the older you are, the more susceptible you are to the becoming confused or delirious. Also turns out the older you are, the more susceptible you are to pneumonia. Another reason not to smoke. Okay. All right, so review your anatomy, especially as it pertains to infection, okay? Now we will review the digestive system. So you could review each of these by looking at your chapters in your book, as well as the links that I provide you, okay? So your digestive tract, as you recall, is a one-way tube that runs through the body. It goes from your mouth, right, allows things from the outside world to pass fully through the body without ever actually entering the body. The only access to the body is what your body allows through the intestine. We have a few things are absorbed through the stomach. Most of our absorption takes place in the small intestine, and then most of our water is absorbed in the large intestine. But we are controlling, right? Our cells are controlling what we take from the food, what's allowed into the body, right? And so when you eat, you're not actually putting that food directly into your body. Although I know it feels like it because it's within, but it is. You have to think of the digestive tract as a tube that goes all the way through. Okay, and that makes sense. We don't want to expose our blood, which would then expose our entire, all of our vital systems, organs. We don't want to take that risk and have toxins and pathogens having free reign in the body. Okay, so recall that the digestive system includes teeth, right? The oral cavity, the tongue. Digestion starts in the mouth with saliva. You have enzymes such as amylase, right? That starts digestion. And then that goes all the way down the esophagus, and of course things are kept going one way through peristalsis of the muscles, and then that goes into the stomach. Where's my pointer? There we go. We got the stomach, and then that will drain into the small intestine, okay, and it's showing the first part of the small intestine, the duodenum, and it's also showing how your pancreas is kind of hiding a little bit behind and a little bit underneath the stomach. And your liver, of course, is above, and the gallbladder right underneath, right? And of course, the bile that your liver makes is stored in the gallbladder. So some can go directly from the liver, as you see, into the stomach. Whereas the excess from the gallbladder can release it into the stomach as well. This is why people can have gallbladder surgery, have the gallbladder removed, because you do have a direct line from the liver to the stomach, but that is going to limit your ability to digest fat. And I really shouldn't have said stomach because it's really going into the small intestine. It's not going into the stomach. So the stomach is about here, and then the liver and the gallbladder have a direct line into your small intestine, the duodenum. Okay, so as I was saying, if you have your gallbladder removed, it's going to make it much more difficult to digest fat. It's not impossible, but you are limited. Okay. The high acidity of the stomach offers a lot of protection. It will kill most microbes, but some microbes will still be able to make their way into the small intestine. What else did I want to say? The liver is super important for being able to detox chemicals, like all the blood that comes from your digestive tract gets filtered through the liver before it's allowed to go into the body. Another level of protection, right? Okay. Brief overview of the intestinal structure and function link is provided for you. And what did I put here? Right, the liver filters blood from the intestines. It also secretes bile, which is stored in the gallbladder. Yeah, I already said all of that. Okay. So the lower digestive system is where most of the time when we're talking about infections, we'll be talking about the small intestine or large intestine. Sometimes you do get a reaction from your stomach, but you don't usually have an infection of the stomach. Okay, so infection, remember, means growth, colonization of a pathogen. Incubation can be pretty quick, it could be as short as 12 hours, but it can also be very slow and take around 2 weeks. When you have an actual infection, you will have a fever. But as you know, sometimes you eat something bad and you pretty much immediately feel it within an hour, up to 48 hours after you eat it. And that would be due to a toxin that was produced by bacteria that you ate. And even if you cook the food, once that toxin has been produced, it's not going away, which is why it's really important to have proper food handling so that bacteria never produce toxins and cause this food poisoning. Okay, so food poisoning is the term we use to refer to the toxin rather than an infection. Okay, gastroenteritis is a generic term, right? Itis, inflammation, irritation of any part of the gastrointestinal system. But typically you will have diarrhea and or vomiting. Okay, if you have really watery diarrhea and if you have bloody diarrhea, then it's called dysentery. Okay, so dysentery is a very severe form of diarrhea, which is not common. Okay, and is very life-threatening. You guys are probably too young to know about the Oregon Trail. One of the first games on computers, but that was it was a game, and your goal was to be able to make it across, right? To make it from one end to the other on the Oregon Trail, and they would constantly people would die from dysentery, and I never even knew what that was until probably college, and I had that a flashback to being a kid and playing that game, and I was like, wow, I can't believe they were talking about that in a kids game. But yes, so dysentery, very severe form of diarrhea, very deadly because it can lead to very extreme dehydration very quickly. Sadly, 1 in 4 children under the age of 5 die in developing countries due to that dehydration from diarrhea because they don't have access to safe, clean drinking water. We have learned that treatment with zinc and rehydration therapy, getting those electrolytes to these children, can greatly decrease their likelihood of dying. But of course, that means, those children would have to have access to this rehydration therapy with zinc, which most of them are not, unfortunately. If you have been intrigued by this idea of clean water, there are charities, foundations that you can donate to help provide clean drinking water in other countries around the world. And feel free to reach out to me if you're interested in the one that I donate to. All right, so most of the intoxications that I talked to you about, these food poisonings, are caused by a specific bacteria, which I mentioned the other day, Staphylococcus, right? So Staphylococcus is the number one cause of food poisoning. That's the ingestion of the toxin produced by the Staphylococci bacteria. Once the Staphylococci bacteria are in the food, they start to release that toxin, and no amount of heating or cooking that food is going to get rid of that toxin. The food is spoiled at that point. But you don't necessarily see or smell anything at all with the food. So good hygiene, washing your hands, using clean utensils, clean cutting boards, keeping everything cold until you're ready to use it and cook it, and putting things away promptly into the fridge once you're done eating them. Very important. Okay, you want things to be kept very hot or very cold. Anything in between is the perfect environment for bacteria to grow. Okay, the urinary system. You know that the urinary system contains 2 kidneys, a ureter from each kidney that goes to the urinary bladder, and then 1 urethra from the bladder to outside the body, and that's the urinary tract. Okay. Infections typically are prevented because we have valves that keep urine flowing one way, and that urine, there's properties of the urine in addition to just the mechanical flushing of the urine that help to get rid of bacteria that have started to colonize the urethra. So this is why staying hydrated and having frequent urination is important for your urinary system. Of course, staying hydrated is important for your whole body. Kidneys, as you know, are filtering all the blood. So basically, think of this at the other end. So you have the digestive system. You're absorbing nutrients and things from your food. It goes through the kidney to get filtered. Then it goes through the entire body. And then at the end of that, it goes through the kidneys to get rid of waste. Where's that waste coming from? It's coming from all of the tissues in your body that have been making waste products as they live, right? A process called metabolism. So all of the processes of the cell that keep the cell alive will lead to waste products, which then go into the blood and get filtered out by the kidneys. And then some of the waste, the carbon dioxide, for example, will then go to the lungs and get exhaled. Okay, so kidneys help to excrete waste. They also help to balance fluid, so you have the right amount of fluid in the blood. You need it to be, you need your blood to be at the right pressure for fluid exchange to happen. There's so many things that you learned in physiology, right? You regulate blood pressure, regulate the amount of red blood cells, and all blood cells. 1/5 of the fluid of your blood goes into a kidney, but much of it is returned right back, right? So you filter it and it goes back. Only a very small amount of molecules actually get filtered out and become urine. And again, like I said, physiology covers this in great detail. Okay, so infections. If you have dysuria, this is difficult and painful urination. Often you also have an urge, like you need to go to you need to urinate often, even when you have already emptied your bladder. If you have cloudy urine, that's pus in the urine. This is from having a surge of white blood cells attracted to that area to get rid of the bacterial infection. And then, of course, some of the cloudiness would be the bacteria themselves, and that's called pyuria. All right. We already talked about the anatomy, so this is just, I don't know, a little review. I doubt you forgot any of this. But one thing I do want to point out is the enormous difference in length of the urethra in a male compared to, I can't even see it in this picture. Here we go, right here. Very short and very long. So women are much, much more prone to urinary tract infections. It doesn't take very long for bacteria to work their way into the bladder. So next time you think to tease women about having to urinate frequently, think again. It's very, very beneficial to urinate frequently and prevent UTIs. Okay. Another really important way to prevent UTIs is good hygiene. This means urinating after sex. It doesn't have to be like the instant afterwards, but do not go to sleep or postpone too long. Okay, always wiping from front to back. Never re-wipe with the same cloth or paper. Never go in the wrong direction, right? Urinating frequently, like I've mentioned, staying hydrated, wearing cotton clothes, which will stay dry, breathe better, avoiding things that are super tight and rubbing. So that's actually a lot of females are giving themselves UTIs because of the clothes that they wear, and also not staying hydrated. And also, perfumey products can cause irritation. This could be from a bath or whatever, okay? So lots of practical things for you for good hygiene practice to prevent UTIs. A UTI is also, a very general term. It doesn't tell you which part of the urinary system has the inflammation, and it doesn't necessarily tell you that it's a result of an infection. When we say cystitis, urethritis, or pyelonephritis, technically means inflammation, right? But we would do a test, a simple test where you urinate in a cup. We use a little dipstick and we measure the amount of white blood cells and an enzyme that's made by white blood cells to determine the amount of, it's indicative of the amount of bacteria that you have. But then we also we do a culture of the urine to see how many bacteria grow. So we do the instant test and we do a confirmation test. And that would tell us if you have an infection. And by the amount of bacteria, and also likewise the amount of white blood cells, that would also indicate where. So if it was simply the urethra, it would be a very low number. If it's the bladder, cystitis, it's going to be a higher number, and then the highest number is going to be if it's in the kidney. So here's an image down here that shows you what the bladder normally looks like, just healthy. You can see all the blood vessels, and a bladder that's inflamed due to intestinal cystitis, right? Inflammation of the bladder due to an infection. You can see it looks very different. You have all these spots, right? It's very spotted, dark spots, thick, dark spots, okay? In the kidney, you can have pus and inflammation in those pyramids, which you remember is where the nephrons are, and that's where the filtration happens. So it's also where it can have easy access to a lot of nutrients from the blood. So, this is like hitting the jackpot if you're bacteria. Okay. A little bit more. We'll see a little bit more about this when I go over specific bacteria. So the prokaryotes are broken into groups, as you know, and we can do this with testing of the what we call rRNA signature, and that will tell us if it's peptidoglycan or pseudomirin. Of course, you could also take, if you have a sample, you can do a gram stain and figure out, from there. Then you could further determine if it's gram positive or gram negative. I said that this is gram negative, gram positive. Gram negative can be divided into a few groups. The major grouping proteobacteria and non-proteobacteria. Gram positive, on the other hand, are broken into two groups called low GC content and high GC content, and that's looking at the amount of G and C in the DNA. And so we'll see this. This branching diagram, notice that it's always an either or, yes or no option. This is called a dichotomous key, and this is very important when trying to determine the identity of an organism. So the first group that we're going to look at will be the gram-negative group phyla called Proteobacteria. The Proteobacteria is such a large group that it's further broken into classes called Alphaproteobacteria, Beta Proteobacteria, Gamma Proteobacteria, and Epsilon. There's actually more, but these are the most medically significant examples that I want to talk to you about. Proteobacteria, the name Proteo comes from Greek mythology, Proteus, meaning a great variety of morphologies. So we see a lot of different types of bacteria. We're going to see intracellular bacteria. We're going to see different shapes, sizes. The only thing that they all have in common is that they're gram-negative. Okay? We might not even get through all of this today because we're already at 48 minutes for this lecture. I want to keep the lecture to about an hour 20 to an hour 30, just like we would have in person. Okay, moving on. By the time you're done with your survey of bacteria, by the end of next week, we will have made it through this entire list. Okay. So this is a really good first step to memorize. So the Alphaproteobacteria, you want to remember that you are in the domain Bacteria, you are in the phylum Proteobacteria, and then now in the class Alphaproteobacteria. These are interesting in that these bacteria have stalks and bud shapes. They kind of almost resemble fungi in this regard. Most are capable of growing at incredibly low levels of nutrients. Okay, and the selected examples that fall into this class are, we're going to look at Bartonella, this is the genus name, Brucella, and Rickettsia. Bartonella are known for cat scratch fever. Brucella are known for undulate fever, not quite as common, and Rickettsia are known for the spotted fevers, and there's a few different species that give the specific disease. So we're going to look at those because there's 3 that are very medically significant. Okay. So Bartonella, most cat scratch fever is caused by Bartonella henselae, and it can cause, this is a very large cyst, enlarged lymph node here. It's not always going to be that obvious, that large. And in fact, some people don't have very pronounced symptoms at all. Now, I do want to tell you, I don't want you to become afraid of cats, or dislike them even more than you might already. If cats have fleas, then there's a chance of cat scratch fever. So you simply need to take good care of your cat, whether it's indoor or outdoor cat. Take it for, its yearly vet checkup. Make sure that it doesn't have fleas. If it does, that you promptly treat the fleas, because it's the fleas that actually have the bacteria, and they excrete it in their feces. And if you're around the cat, that feces can then get on you. And if you have any scratches, or if the cat were to scratch you, such as just playing around, then that could introduce the feces from the fleas into your wound. And of course, the bacteria would be in the feces of the fleas. And so, kind of shows you that our diseases can have rather complex pathways, life cycles. Okay, so fleas can transmit Bartonella, and we also have lice and sandflies that can transmit this other species called Quintana, which isn't so popular today, but was a huge problem during World War I, and it was called trench fever. Body lice infestations were a huge problem during World War I, and so lots of soldiers came down with trench fever. But getting back to cat scratch fever, so you can have fever, muscle pain, enlarged tender lymph nodes. It can develop from 1 to 3 weeks after exposure, and typically wherever the inoculation site is, whatever wound the flea feces got into, will have some type of like pustule, so blister. It's rare, but it is possible to have complications where it can be anywhere from eye infection to very severe muscle pain, and even encephalitis and endocarditis have happened. But like I said, this is pretty rare. It's difficult to detect this pathogen because it is an intracellular pathogen. So this bacteria, once it gets into your system, it will actually infect. It will go inside of your cells and infect the inside of the cell. Okay, and very interestingly, it can actually form a biofilm in the blood vessel, and this is incredibly rare. We typically don't find biofilm infections unless there's some implant of some sort, something artificial in the body. So this is an exception to that rule here. But because it's an intracellular pathogen, it's very difficult for us to detect it, and it's equally difficult to treat. How do you get the antibiotics into our cells and then into the pathogen? It's very well protected. The pathogen, intracellular pathogens, are hard to treat and hard to detect. Okay, so one more thing to tell you about Bartonella. It is a bacilli, rod-shaped bacteria. I already told you it's intracellular. Another thing is that you can develop a rash that would be a streaked pattern. Okay. And we really have to do something like a serological test and also PCR to diagnose it, because it is difficult to diagnose. So serology is using antibodies. PCR would be trying to do a DNA extraction. So taking a sample from the patient, extracting the DNA, and then amplifying the DNA to specifically detect the pathogen's DNA. To treat it, you can do something like azithromycin, erythromycin, or doxycycline. These are the 3 antibiotics that are used to treat Bartonella infections. Okay, next. Brucella. This is more common for people who are working with cattle, sheep, goats, pigs, or wild game. So you can imagine the most common would be people that are working in dairy or meat factories, um, where they have direct contact with the mucous membrane. Um, right, so they touch the animal and they then might touch their eye or their nose, or eat something and have it get in their mouth. Okay, and initial symptoms, just having a prolonged fever. You might have fatigue and muscle joint pain. And long-term symptoms is just having that recurrent fever and chronic fatigue. That's really usually it. But sometimes the heart, liver, and spleen, even if it's a man who's infected, testes and scrotum can even become swollen. Of course, the swelling of the heart, liver, and spleen are what typically get the attention and diagnosis of this. But going to the doctor with chronic fatigue and recurrent fever would definitely get the doctor's attention. I just don't know how well they would be able to diagnose it until you have some more symptoms to guide them towards this. This, of course, everything we're looking at right now is gram negative, but this is an example of a cocobacilli. It's very small, and it's in between. It's not quite circular, and it's not quite rod-shaped, right? It's in between. This is also an intracellular pathogen, okay? And the disease, we call it, sometimes we just call it brucellosis after Brucella, and sometimes we call it undulant fever for this recurring fever. Okay, treatment is 6 to 8 weeks on antibiotics because it's very hard to kill this intracellular pathogen. So people who work with livestock need to take careful precautions. Okay, the Rickettsia. These are the spotted fevers. We have Rocky Mountain spotted fever seen here. Its distinguishing characteristic is that you have the rash on the palms of the hands and soles of the feet. And I didn't provide images of the other diseases. Probably just can't fit everything on this slide. Okay, so all of the Rickettsia are also intracellular parasites. So interestingly enough, the 3 examples that I have selected to represent the Alphaproteobacteria have been intracellular parasites. I don't want you to think that all Alphaproteobacteria are intracellular. They're not. But it turns out that the most medically significant examples that I wanted to show you are. So they're obligate intracellular parasites. They have to get inside of the host tissue, and not just in the tissue, but in the cells themselves. They can induce phagocytosis and survive inside the phagocyte. You can get these from arthropod. But you can also get these, so here we'll see lice feces, and then here we see rat flea feces, and then here we see ticks. So the arthropod borne is for Rocky Mountain spotted fever. You get it from a tick, and then that's how you get it. Okay, Rocky Mountain spotted fever is the most dangerous. Well, they all have a high mortality rate. They're all very dangerous. But Rocky Mountain spotted fever has to be treated within the first 5 days, or else treatment will not work. Okay, Rocky Mountain spotted fever is caused by the species Rickettsia. So Rickettsia, kind of fun to say. And that's shown here. So all these dark red spots are the bacteria, okay? And you can see they're inside of the host cell. And this would be the nucleus of the host cell, okay? Also seen here. Tissue damage as bacteria multiply. in the endothelial cells. The cells burst open, and then this can disseminate to the brain, liver, heart, and kidney. So this is why it's really important to treat very rapidly. Going back up to the top, we have Rickettsia prowazaki, known as epidemic typhus. Epidemic typhus is caused by lice feces, and then that is from there goes to humans. Okay, is very high mortality rate of 40%, and another, like, some symptoms, some characteristic symptoms for you include light sensitivity, low blood pressure, severe headache, confusion. And then you have the Rickettsia typhi, which is known for endemic typhus. So you have to be very careful not to confuse these two, right? So prowazaki is epidemic, and so you can remember P for prowazaki and P for epidemic. And then typhi is endemic, also known as marine for rats. So the fleas on the rats, it's the feces of the fleas on the rats. Okay. So rodent control is important. So you might think, I don't know why people are so afraid of mice or rats. They're no big deal. Well, they are a big deal because they can be vectors of very serious disease. So you don't need to scream if you see one, right? You don't have to be that afraid, but you need to make sure that you never have rodents where you live, where you eat. Okay. If you are ever around rodent feces, you need to be careful, and you need to, get a mask, get gloves, and then make sure that you clean up very careful. Okay. All right. That's it for this. So this gives you another view. So I was showing you here the Rickettsia bacteria, intracellular, right? This is showing you the same thing. But here you're seeing it in lab grown in a chicken embryo cell. And then these are just huge masses of Rickettsia in the nucleus of these cells. And then scattered about the cell as well. So just completely overtaken this chicken embryo cell. So Rickettsia grow only within a host cell, right? I told you obligate intracellular pathogen. And the chicken embryo is not the normal host. This was done in a lab setting. Okay. Here we're looking at the actual bacteria now, close up. Okay, this is a tunneling electron microscope. So this is magnified way more than what a microscope in our lab would be able to do. But you can see that they do have a slime layer. We learned about that glycocalyx. And then you can see, so Rickettsia cell that has just been released from a host cell and then imaged. So this is just showing you what it looks like. So another organism in the Alphaproteobacteria group is not actually a human pathogen, but is such a widespread pathogen of insects and other organisms that it is really important to mention. It's also really bizarre and interesting. So it can cause a lot of different outcomes in different organisms. This can range from, for example, it's an endosymbiont. Sometimes it's a parasite, and sometimes it's a mutualistic relationship. In some insects and invertebrates, such as ants, nematodes, mosquitoes, and flies, sometimes if you were to give like an antibiotic treatment to kill the Wolbachia bacteria, the organism will die. So their very survival is linked to the presence of Wolbachia. Other times it's their fertility is linked, and they would not be fertile. And sometimes we see even other more complex interactions with the host. Another example of survival is like nematodes. You kill the Wolbachia with antibiotics, and the nematode worm dies too. And for some insects, the Wolbachia actually turns the males into females and results in unfertilized offspring, and you get all females. I don't know why that's the case. That doesn't seem beneficial, because eventually females die, right? And then, so yeah, it's very bizarre. But this is what we've been learning about studying Wolbachia and different organisms. Super fascinating. You could definitely, I usually don't encourage Wikipedia, but it's been around for so long that it's actually pretty darn reliable, and they always give citations, and you can always read their sources. But you could definitely read about this for fun, because it's so bizarre. Okay, but it does not cause disease in humans, and it actually offers us potential control mechanism if we find the right strain of Wolbachia to either stop or slow down the mosquitoes that spread malaria or other diseases, right? It does offer potential control mechanism. You have to be very careful when you start playing around with nature, though. If you take one organism out, usually another organism will rise to the occasion, and then you have to start all over again. And it could be even more difficult to deal with. So, yeah. Welcome to the world of microbiology. Isn't it fun? Isn't it interesting? So what is this? Another dichotomous key. So you could categorize things as whether or not they cause disease in humans. And then say it does, and say it is an obligate intracellular parasite. Then you could ask the question, well, does it cause spotted fevers? Yes. Does it survive in phagocytes? If it survives in phagocytes, maybe it's Bartonella, maybe it's Brucella. But if it causes the spotted fevers, then you know it's going to be like a Rickettsia. Okay. If it doesn't cause spotted fever, then it's not Rickettsia. It's this other group that we didn't talk about, so you don't have to know anything about this guy. Okay. We will make our way through this side as well. You do not need to memorize these dichotomous keys. I want you to get the hang of looking at them and using them. I want you to know what a dichotomous key is, but there's no need to memorize the keys. You do, however, need to memorize everything else that I've given you in these presentations, both what's on the slide and what I've told you orally, right? Okay, so here's a question. Which one of the following can cause a wide range of human infections, including pimples, boils, pneumonia, food poisoning, and surgical wound infections? Now, this question, I only talked about one of these items today, but I have talked to you about the other items in previous lectures and labs. Hopefully you remember. So food poisoning we talked about today. Which one of these is capable of causing food poisoning? That's right, Staphylococcus B. Staphylococcus, and specifically Staphylococcus aureus. And you'll realize if you haven't already memorized this guy, you will today, right? Staphylococcus aureus is a huge pathogen, causes all sorts of disease. All right, so that's our answer, and I think that's where we're going to end today. So look at that. I finished 10 minutes ahead of schedule. Okay, so I will see you guys next week for part 2 and part 3 of Bacterial Survey. Bye.